All right, let's dive right into this explainer and finally demystify the ICH Q14 guideline on analytical procedure development. Look, if you're working in pharmaceuticals or QC, you already know the drill. Keeping your analytical procedures up to date without getting completely bogged down in endless regulatory filings, that's basically the holy grail, right?
So this brings us to the million-dollar question for, well, pretty much every lab manager and regulatory affairs specialist dealing with those super rigid testing frameworks. How exactly do you ensure a drug test is perfectly fit for its intended purpose, but also future-proof? Well, today we're going to see exactly how ICH Q14 answers that, basically by shifting us away from those rigid, static methods and moving toward a much more dynamic, life cycle approach.
Here's a quick look at our agenda for today. We'll be hitting the goal of ICH Q14, minimal versus enhanced approaches, the analytical target profile, parameter ranges and robustness, control strategies and changes, and finally, multivariate analytical procedures.
Okay, Section 1. The goal of ICH Q14. Designing for the entire life cycle.
Traditionally, modifying an analytical procedure post-approval has just been a massive headache. You want to upgrade a piece of equipment or maybe just tweak a method, but that looming regulatory gridlock makes you second-guess if it's even worth the effort. But Q14 introduces a really systematic, science-based escape route from all that. It's all about designing a procedure that's flexible from the start and understood so deeply that future changes are actually manageable. And this roadmap really beautifully illustrates that entire analytical procedure lifecycle. Notice how your prior knowledge and your risk assessment feed directly into your control strategy and those post-approval changes. Basically, your product and process understanding inform exactly which attributes require testing. And then that feeds right into your technology selection, risk assessment, and parameter identification, which leads all the way down to routine use and continual improvement. It's just a continuous, highly logical loop. Moving on to Section 2, minimal versus enhanced approaches. Choosing your development path. Now, the minimal approach remains perfectly valid, focusing on identifying attributes, testing accuracy, setting fixed parameters, the usual stuff. But what's truly interesting here is how the enhanced approach systematically builds profound procedure understanding. The enhanced side utilizes rigorous risk assessments, multivariate experiments, and it really paves the way for genuine life cycle flexibility down the road. Which takes us to Section 3, the analytical target profile, defining the goal before the tool. So the analytical target profile, or ATP, is basically a description of the intended purpose of the analytical procedure, along with the relevant performance criteria for the product attributes you need to measure. Think of the ATP as writing a super strict job description for your test before you even decide what specific machine or what technology you're going to hire to do the actual work. Because by establishing this profile early on, you're creating a really sturdy foundation that ensures your analytical procedure remains fit for its intended purpose throughout its entire life. The ATP actually drives your technology choice, because let's face it, multiple technologies might actually meet those criteria. This upfront clarity facilitates ongoing monitoring and just makes continual improvement a whole lot smoother.
Section 4, parameter ranges and robustness, navigating challenges and setting boundaries.
When it comes to challenges, quality risk management, or QRM, is absolutely your best defense. By utilizing tools like Ishikawa diagrams, you can actively reduce the risk of poor performance. It helps you prioritize exactly which parameters actually need experimental testing. And in turn, this informs your ongoing monitoring and keeps your whole procedure robust over time. All right, we're definitely getting into a bit of the regulatory alphabet soup now, starting with PAR. A proven acceptable range is established by the univariate examination of a single analytical procedure parameter. Simply put, a PAR defines the safe operating boundary for one specific parameter. But taking it a step further through multivariate experiments, we have the MODR, or Method Operable Design Region. An MODR shows how multiple parameters interact safely together. So it's not just about turning one dial on the machine. It's about understanding how adjusting multiple dials at the exact same time still keeps you in a totally safe, validated, and operable space. So why does all this matter for your lab? Well, because if you stay within these approved boundaries, whether we're talking about a PAR or an MODR, you bypass the need for tedious regulatory notifications. This is absolutely huge. It literally gives your lab the flexibility to adjust operational conditions for normal, day-to-day variances without triggering an absolute mountain of compliance paperwork.
All right, Section 5, Control Strategies and Changes, reaping the rewards of the enhanced approach.
Let's talk about established conditions, or ECs. These are the analytical procedure parameters that absolutely must be controlled to ensure performance. But here's the kicker. By using the enhanced approach, the number of rigid ECs can actually be reduced. You can focus them strictly on true performance criteria, rather than just arbitrarily locking down every single minor detail of the procedure.
This decision tree elegantly maps out how proposing ECs based on high, medium, or low risk dictates whether a future change is going to require prior approval or maybe just a simple notification. If you have sufficient understanding to design bridging studies, a change that might normally require a really lengthy prior approval could potentially drop down to a moderate or even low notification.
So there's a very direct relationship here between your level of knowledge and the risk of a change. When you have high procedure understanding, meaning high knowledge, even high-risk changes can be managed with studies derived safely from prior knowledge. You aren't just starting from scratch every single time you need to adapt.
The ultimate payoff here? Fewer regulatory hurdles and the seamless implementation of new technologies way later in the life cycle. Putting in that enhanced work up front acts like a regulatory insurance policy. It basically guarantees immense, long-term flexibility for your lab. Finally, Section 6, Multivariate Analytical Procedures, Additional Considerations for Complex Models.
Because these models link measured variables with reference values, having a robust sample selection that captures real-world commercial-scale variability is absolutely critical. You also have to carefully select variables, like wavelength ranges, and you really have to balance the trade-off between accuracy and robustness when choosing your latent variables. Plus, utilizing outlier diagnostics is pretty much non-negotiable for ensuring your data quality.
You really can't just build a model and forget it. Ongoing outlier diagnostics and event-driven confirmatory testing are completely essential to maintain compliance and to ensure that your calibration model actually remains reliable over time.
Which brings us to our final thought. Look, the minimal approach to procedure development is entirely valid, for sure. But is it really worth the long-term inflexibility? As you evaluate your own analytical procedures, you've got to ask yourself, if investing in the enhanced approach today will save your team from the regulatory gridlock of tomorrow, which path will your lab take? Thanks for exploring this with me today, and I really hope these insights empower your next steps in procedure development.